So my name is Jesse, and I'm going to be talking about determinants of human versus mosquito cell entry by the chikungunya virus envelope. And this is the first time I've given this talk. Actually, about 9 months ago, I was giving a seminar at a university, and I was talking about SARS-CoV-2 evolution, which is mostly what I've talked about for the last 5 years. And after the talk, one of the grad students at the grad student lunch said to me, "You know, that was a pretty nicely done talk, but what are you going to do now that nobody cares about SARS-CoV-2 anymore?" So I decided I better make a new talk for this symposium. Okay, so I'm going to be talking today about chikungunya virus. So chikungunya virus is an alpha virus. It's a mosquito-borne virus, and although it's very rarely fatal, it causes really severe joint pain and musculoskeletal symptoms during infection. So it's a very painful virus to be infected with, and some of these symptoms can, in some cases, persist long after the infection has been cleared. And it's transmitted by both Aedes aegypti and Aedes albopictus mosquitoes. So new outbreaks with chikungunya virus have been sort of expanding around the globe, and this is driven in large part probably due to climate change-induced spread of the mosquito hosts to larger geographic regions. So regions where these mosquitoes might not have been present decades ago, they're increasingly expanding their host range. In addition, there have been probably some adaptations in the virus over the last few decades that may have expanded its ability to infect additional mosquito species. So for instance, I don't know how many of you read this really interesting article in the New York Times a couple weeks ago about how there's a large outbreak going on in a region of China right now, and it was describing all the things they were doing, including introducing these elephant mosquitoes, which eat the other mosquitoes, and using drones and all of these things to try to get rid of the mosquitoes. There's a number of regions in North America, including the US, where expansion of these mosquito species is also going to make this virus an increasing risk. So as part of its natural transmission cycle, chikungunya virus infects both mosquitoes and human hosts, as well as some non-human primates. So it's an arbovirus. So basically, the mosquito is infected, the mosquito bites this host, the virus is now replicating in the mammalian host. Another mosquito comes and bites that mammalian host, and that's how the virus circles. And there can be this sylvatic cycle where this is happening in non-human primates, but a lot of the transmission is in an urban cycle where the non-human primate is a human primate, namely a human. And so as with all arboviruses, so viruses that are able to infect both mosquito and human hosts, this virus has to be able to enter the cells and replicate in both of these different species. And for any of us who think about virus host tropism, I always think that these sorts of viruses are pretty remarkable because for many viruses like influenza and SARS-CoV-2, we study the adaptations that are needed even for the virus to go from one mammalian species to another mammalian species, and often that requires a lot of adaptation. But here are these viruses that are able to infect these much more highly diverged mammalian and invertebrate hosts. So the way that chikungunya virus gets into the cell, which is what I'm going to be talking about today, is via its envelope proteins. So in the viral genome, there's a number of genes, or these non-structural proteins, which are essentially the genes that are amplifying the viral genome and doing other functions unrelated to cell entry. And then there's the structural proteins, which are synthesized as a single, very long polyprotein, which is then cleaved into capsid, E3, E2, 6K, and E1. And I'm going to be referring to the E3 through E1 region as the envelope proteins, because E3 and E2 and E1 form the actual sort of physical envelope on the surface of the virus, and then 6K interacts with that, although it's not actually part of the physical structure. And so in mature virions, it's only E2 and E1 that are left as part of this polyprotein. E3 is cleaved off. And so there's these trimers on the surface of virions, as shown here, and they're sort of densely packed on the virion. And so roughly speaking, although there's not a 100% delineation between these, E2 is the primary receptor binding protein, and E1 is the fusion protein. And so this is a type 2 fusion protein, unlike the type 1 fusion proteins that people may be familiar with for something like the SARS-CoV-2 spike or flu hemagglutinin or HIV envelope. And so, if you just look at the number of amino acids here, these envelope proteins, they're quite large when you look across all of the proteins together. And so about 6 or 7 years ago, Mike Diamond's lab at WashU was able to identify the human receptor, or at least one of the human receptors. I wouldn't say it's completely proven this is the only human receptor, but it's at least an important human receptor, which is a protein called mixer-8. And so the binding of mixer-8 to the envelope proteins is super complicated and always gives me a headache when I look at it. I spent all this time working on SARS-CoV-2, where there's a little receptor binding domain that binds onto ACE2, and you can kind of understand what the interface is. So here's a structure of the envelope protein's trimer, and that trimer binds to multiple mixer-8 molecules, and the mixer-8 molecules sort of wrap around one of these E2 proteins, but then also make contacts with another E2 protein in the same spike, and actually it's not shown here, but there's also sort of another adjacent trimer that's also making some contacts here. And then it also contacts E1 to some degree. So there's a binding interface here, but it's sort of a complicated binding interface to visualize because on any given monomer of E2, it's interacting with multiple different mixer-8s in different modes. But there's been a lot of nice work by Mike Diamond's lab and others showing that this mixer-8 is quite important for the entry of this virus into human as well as mouse cells. And so what I want to talk about in the talk today, and sort of the goal of this research, is to define the host-specific sequence determinants of cell entry. So we have no idea what is the receptor in mosquito cells, but it's definitely not mixer-8 because there's no mixer-8 or even distant homologue of mixer-8 in the mosquito genome. And so in the absence of knowledge of what that mosquito receptor is, one of the questions we wanted to ask is, can we understand how mutations to these viral envelope proteins affect the ability of this virus to infect different types of cells, including human and mosquito cells, and can that teach us something about how it might be entering these cells? So the approach that we've been using to do this is what I'm going to refer to as pseudovirus deep mutational scanning. So I'm going to give some details about how this works in a second, but first I just kind of wanted to give an overview. So the key idea, before you read any of the text, is we're just making a big library of different virions that have different envelope proteins on their surface. So there's the yellow indicates one mutant of the envelope protein, the green indicates another mutant, the blue another mutant, and so on. And we make so many of these that all of the different possible amino acid mutants of these envelope proteins are on the surface of one of these virions. And so we do this in the context of lentiviral particles, so pseudotyped lentiviral particles, which I'm going to refer to in the rest of this talk as pseudoviruses. And these pseudoviruses, in addition to having a different envelope protein on their surface, they also have a little barcode in their genome which tells us which envelope protein do they have on their surface. And the reason we do that is because, as I just described, these envelope proteins are large. The gene encoding them is over 3 kb long, and we don't want to have to sequence that thing. So we want to just be able to sequence this little barcode and know what mutant is on the surface of this. And then an advantage of doing this in this pseudovirus context, as opposed to doing it in the context of something like actual chikungunya virus, is these pseudoviruses can only undergo a single round of cell entry. So probably most people here have used pseudotyped lentiviral particles for something in their research. They're extremely widely used in biology, and they can enter cells once because you produce them in a way that they have the viral proteins, but then they can't enter cells again. So they're not human pathogens, which makes them an attractive way to study mutants of viral proteins at biosafety level 2. So more specifically, how do we do these experiments? So here's our lentiviral genome, or our lentiviral backbone. So just like a lentiviral vector you would use for anything else, there are these non-coding regions at the termini. There's a 5' LTR, some other important lentiviral elements. And then at the 3' end, there's a repaired 3' LTR. And so this is important because it allows the lentiviruses to be reactivated. Many lentiviral vectors that people use in the lab have deleted 3' LTRs. And then in this lentiviral genome, what we've done is we've put the chikungunya virus envelope polyprotein under control of an inducible promoter. Then we put a little stop code on, and then downstream of that, we put this little nucleotide barcode, which is just 16 random nucleotides which is going to lead to a different barcode sequence being associated with every envelope protein's mutant. And then we also have some fluorescent reporters and antibiotic resistance markers in here. And so these lentiviral genomes don't encode any viral proteins other than these envelope proteins. And so they can't undergo multi-cycle replication for that reason. So then what we want to do is we want to use these backbones to create that library of genotype, phenotype linked lentiviral particles. And this actually turns out to be a lot more complicated than you would think, and I'm going to walk through why it's complicated. But the key reason that this complicated process is needed is because of two features of sort of these types of experiments. So first of all, as most people are probably familiar, when you transfect a mammalian cell with plasmids, it takes up, if it takes up any plasmids at all, it takes up lots of plasmids, like tens of thousands of plasmids. This is different than when you transform a bacteria, right? When you transform a bacteria, they usually get 0 plasmids or at most 1 plasmid. So if you transform a bacteria, you kind of automatically have a genotype-phenotype link. When you transfect a mammalian cell, they take up a whole bunch of things at once. So if you transfect in a whole bunch of different viral protein mutants, each of these cells is making a whole bunch of different viral protein mutants at once, so there's no genotype-phenotype link. And then the second thing is that lentiviruses are what are called pseudodiploid. So they actually package 2 genomes, or have diploid genomes, even though they only integrate one of those. So what that means is in order to make these genotype-phenotype linked viral libraries, we can't just do what would be simplest, which is transfect these backbone plasmids along with the standard lentiviral helper plasmids that encode the other HIV proteins into 293 T cells. Because if you do that, you get out a mix of viruses that are packaging 2 different genomes and may have, God knows what mix of envelope proteins on their surface. So instead what we do is we transfect these things just like we're making a VSVG pseudotyped lentiviral particle as you might be doing for, let's say, gene delivery. And then what comes out is a library of VSVG pseudotype virions. So they now have this vesicular stomatitis virus glycoprotein that can infect any cell on their surface. And each of them is packaging 2 different ones of those genomes I was just showing before, which has some envelope protein mutant and barcode. We then take those and we infect them into 293 T cells that are expressing this protein RTTA, which is needed for the inducible promoter, at low multiplicity of infection. So most of these cells don't get any viruses integrated in their genome. And then because it's low multiplicity of infection, most of the ones that get anything integrated in their genome at all just get 1 copy. And then we just enrich for the ones that have 1 copy. So now what we've done is we've essentially taken this lentiviral backbone that I just showed here on the previous slide and stored them 1 backbone per cell in this library of cells. So this viral library is now sort of stored 1 copy per cell in these cells. And now we can easily generate these genotype-phenotype linked viral libraries by just transfecting the other HIV helper plasmids. And then you get out viruses that have each has a different envelope protein mutant on its surface. And if you want to be able to get out viruses that sort of allow you to figure out the composition of this library independent of whether that envelope protein is functional, you can also transfect in VSVG. And then they have VSVG on their surface. So they're all sort of functional for entering cells. And then the last key thing, if anybody tries to do this, is and people who worked in single-cell CRISPR screens know about this stuff as well, because it's also a bugaboo there, is that we do the long-read sequencing, which links the full envelope protein variant to the barcode after we've done this integration step, because what's in the plasmids can get recombined relative to what's in the cell. But once we've done that here, we now have each of these envelope protein variants on the virus associated with the unique nucleotide barcode which is in the virus. So we can now produce these viruses, infect them into cells, and do very cheap, short-read Illumina sequencing to figure out which viral mutants we're able to get into any cell. So that's how we make these libraries. So we can now use them to what I'm going to describe here. First, measure how each mutation to these envelope proteins affects its ability to enter mixer-rate expressing cells. So we use those cells to first just generate VSVG pseudotyped virus from those cells. So that sort of allows us to tell what is the distribution of things that were in those cells in the first place. And then we can just take those, infect them into 293 T cells expressing mixer rate, and all of them will be able to affect 293 T cells because they have VSVG on their surface. And we can sequence the barcodes to figure out what was the relative frequency of all of these things that we produced in this library. But we can now also produce them without VSVG. And now they can only enter the 293T mixer 8 cells if the envelope protein mutant they have is able to enable a virion to enter 293T mixer 8 cells. And so we sequence those barcodes. And so if you got a pattern like this, you would, for instance, say that this orange mutant is an envelope protein that's not very good at entering 293T mixer 8 cells, because you see a lot less of it in that condition than you did in the condition where it could just use the VSVG to get in. So we have these libraries that have essentially all of the envelope protein mutants, and we do this experiment. So first we can just look at a very coarse level for envelope proteins with different types of mutants. How can they enter cells? So we quantify that entry of cells with a cell entry score, where 0 indicates that envelope protein mutant infects the cells just as well as the wild type, or unmutated chikungunya virus envelope protein, and a negative score indicates it's worse at entering the cells. So you can see the envelope proteins that have no mutations or have only synonymous mutations. For all of those, and there's tens of thousands of different barcoded variants like this in these cells, we all measure a cell entry score of around 0. There's a little bit of spread because there's some noise in these experiments. The ones that have a single amino acid mutation, some of them are still entering cells about like the wild type, and some of them are much worse at entering cells. And then the ones that have multiple amino acid mutations, more of them are worse at entering cells, because the statistics of that is sometimes mutations are bad for cell entry, and usually you put several mutations together and you end up even worse than if you just had one mutation. And then the ones that contain stop codons are uniformly bad at entering these cells, because if you truncate the envelope proteins, they don't work. And we have 2 different libraries, although throughout the rest of this I'm just going to be showing the average of the measurements across those. So this is sort of aggregated at a high level, but the cool thing is that we can actually go and look at all of the individual mutations. So here is a big heat map where each point in this heat map is a different mutation, and the color is its effect. So this is like the mutating site 7 in E3 from an M to a G. And it's colored red because that mutation is bad for entering those cells. And there's other ones that are colored white because the mutations have very little effect on entering cells. And since this is a big heat, so you can use this heat map to look up any specific mutation you're interested in. Since it's a big heat map, it's kind of hard to look at. So we can also summarize it in this line plot, which is just showing the average effect of mutations at each site. So you can see if we look in the E1 region, mutations are somewhat deleterious, but many of the mutations are pretty well tolerated. But we can identify regions, such as this region in E2 here, where a lot of the mutations are really bad. If you go to this 6K protein, which is sort of this accessory thing which is not essential, at least for entry in cell culture, you can see this is the 6K region here. The mutations are all pretty well tolerated. And then there's certain mutations like this part right here of E1, which is really largely involved in the fusion peptide, which is a really important part of this protein, where the mutations are basically all really bad for cell entry. So there's this sort of huge data set we can look at if you care about specific mutations. So one of the things that's nice to do is just sort of project that information onto the structure of the envelope proteins. So here's the trimer of the envelope proteins showing E1, E2, and E3, and here's where one of the mixer 8 molecules bind. And each monomer in the trimer is colored differently, but in all cases, a darker color means the mutations are bad for cell entry. So you can see the part of E2 that contacts mixer 8. If you make mutations there, that tends to be bad for cell entry because it can't bind to cells anymore. And there's also regions of E1, particular regions that correspond to this part of this protein that are very important for sort of the membrane fusion where mutations are quite bad. And we can, if this thing actually manages to load here, let's see if it does, you can even kind of call that out in more detail. So here's the structure of mixer 8 shown here in yellow with the envelope proteins. And if you look at these sites where mutations are really, really bad for cell entry, like the ones I'm clicking here, they tend to be sites that are in close contact to the envelope protein when they're in E2. So this all sort of makes sense that we sort of defined one of the things you would expect, that if you mutate the parts of this protein that are important for membrane fusion, it doesn't work anymore. in terms of entering cells. If you mutate the parts that are important for binding to mixer rate, it doesn't work very well for entering cells. So then we wanted to ask, all right, how do mutations affect entry in different cell lines? Because we already know when you take these human cells, these 293 T cells, and you make them express mixer rate, the virus is using mixer rate to enter those cells. So we now wanted to compare that cell entry to cells where the virus is entering via binding something that's not mixer rate. So I'm going to discuss two alternative cell lines here. One is called 293T TIM1 cells. So these are human cells that express TIM1. So TIM1 is a protein that binds to phosphatidylserine, which is will end up being a component of just the viral membrane for these. So basically, TIM1 is a factor that promotes the entry of a lot of different viruses into cells, and it's not actually dependent on a specific receptor interaction. What's happening is when your 293 T cells have TIM1, they're just binding to this phosphatidylserine, which is on the membrane of these viruses. So this is sort of a way where the virus can potentially get into cells without having to bind to receptor, at least without having to bind to receptor very well. There may still be some receptor binding that's needed for triggering. And then the second cell line are these C636 cells. So this is an Aedes albopictus mosquito cell line. It does not express mixer rate because mosquitoes don't have mixer rate, and it doesn't express TIM1, but somehow chikungunya virus infects these cells. So if we just look very broadly, here what I'm now showing is I've taken that big heat map I showed you for 293T mixer 8, and I've plotted each point here as a different mutation, and I've plotted the effect of that mutation on entry in 293T mixer 8 cells or one of the other cells. So first, let's just compare this, 293T mixer 8 versus 293T TIM1. So you can see most of the points are on this diagonal because most mutations have the same effect for entry into both of these cells. And this is something that you will always see in this type of experiment, because the biggest way that mutations affect the ability of the envelope proteins to mediate cell entry is they just mess up the folding of the envelope protein. So if you mess up the folding of the envelope proteins, you can't get into any of the cells. But you can see there are some mutations here that are tolerated in 293T TIM1 cells. So they're close to 0 on the y-axis, but they're very deleterious in 293T mixer 8 cells. And if you break down what proteins those are in, you can see those mutations are overwhelmingly in the E2 protein, which makes sense because the E2 protein is the thing that binds to mixer 8 to get the virus into the 293T mixer 8 cells. Then likewise, if you look here, I'm just showing the E2, comparing the 293T TIM1 to the C636 cells, the mosquito cells, you can again see that there's a fair number of mutations that are bad for entry in these mosquito cells, but are pretty well tolerated in these 293T TIM1 cells, which suggests that there's some part of this E2 protein which is having to bind to something on the mosquito cells to get the virus in, but it doesn't need to do it in these sort of hyperpermissive 293T TIM1 cells. And finally, if you compare the human mixer 8 expressing cells and the mosquito cells, you can see there's some mutations sort of on both sides, which could suggest that there are some mutations that are important for entering one cell line, but not entering another cell line. And so another way to look at this, if you just kind of want to call out the specific mutations, and the mutations we are most interested in are the ones that can enter the mixer 8 expressing human cells versus mosquito cells, is like this. So here what I'm showing, negative values mean mutations that are a lot worse at entering the 293T mixer 8 cells than the mosquito C636 cells. So here, this is an example of such a mutation at site 119. Here you can see the heat maps of the mutation effects in all of those cells, and here's just an easy way to look at it. Here's the effect of that mutation in in C636 cells, and here's the effect in 293T mixer 8, sorry, other way around, and C636 cells. So here are mutations that are bad for entry in 293T mixer 8, but well tolerated in C636. So these are candidate mutations we would think would be specifically impairing human cell entry. And here's an example of another mutation like that. And then there are also mutations like this one, which are pretty well tolerated. So like if we look at this mutation, it's pretty well tolerated for entering the 293T mixer 8 cells, but it's bad for entering the mosquito cells. So maybe that's doing something specific in binding the mosquito cell receptor. And so as some sort of sanity checks, although we don't know the mosquito receptor, we know in 293T mixer 8, the receptor is mixer 8. So if we look at those mutations that are worse at entering the 293T mixer 8 than these sort of hyperpermissive 293T TIM1 cells, you can see the mutations that are worse at entering the 293T mixer 8 cells are ones that have a low distance, so are close in the known cryo-EM structure of where mixer 8 binds to the envelope proteins, which kind of makes sense because probably many of these mutations are affecting that. So we don't know the mosquito receptor, but what we can do is we can plot the same thing. So here's the difference in entry on these C636 cells minus these 293T TIM1 cells, now projected onto the structure of one of the monomers of E2 and E1. And mixer 8 is not the mosquito receptor, but it's a mammalian receptor, so I've sort of modeled it in so we can see where that binds. And as I mentioned, any given monomer is binding sort of two different mixer 8s in different modes. So red here are mutations that are deleteriously affecting entry into the mosquito cells. And you can see there's sort of a surface largely on E2, which is shown here, or is easier to see in this rotated view here, which is sort of a continuous surface where there's a lot of mutations that are bad for entry in the mosquito cells. It partially, but not completely, overlaps with the mixer 8 binding interface. So this is a candidate binding interface for the mosquito receptor or receptors. So everything I've showed you so far was in the context of these pseudotyped lentiviral particles, which, as I described, have a number of attractive features for doing these experiments, but they're different than actual alphavirus particles. So to validate this in the context of alphaviruses, we first created what are called these alphavirus reporter particles. So to make these, we transfect cells with a genome for an alphavirus. This is an attenuated strain of Venezuelan equine encephalitis virus, which is a different alphavirus, that has the non-structural proteins, and then it has a reporter. And then we transfect the structural proteins, which are from chikungunya virus, so the capsid and the envelope proteins on different protein expression plasmids. So again, this forms these reporter virus particles which don't package all of these proteins, and so they're not actually multi-cycle replicative viruses. So again, they're safe to study these mutants at biosafety level 2. And when we put those mutations in there, so we chose for these ones that had a variety of mutations that were measured to a specifically decreased entry in 293T mixer 8 cells. Those are colored blue. Or in C636 cells, those are colored orange. So when we look at mixer 8 expressing human cells, so here's the 293T mixer 8 cells, you can see, consistent with the pseudovirus deep mutational scanning, the mutations that are supposed to negatively affect entry in mosquito cells, but not the human cells, have no effect. They have titers similar to the wild type. And the ones that have the single mutants that are supposed to be bad for entry into the human cells are in fact bad for entry into these cells. When we make some combinations of these mutations, they get even worse. And then we partially validated this in HUH7, which is another human cell line which endogenously expresses mixer 8. Conversely, when we go to the mosquito cells, so here I'm showing C636 and also a different cell line, an Aedes aegypti cell line called AAG2, you can see the mutations that were measured to specifically decrease entry into the 293T mixer 8 cells don't impair entry into those mosquito cells, but the mutations that were measured to impair entry into the C636 cells have reduced titers as single mutants, and even more so when you combine them as double mutants. So this is validating in the context of these alphavirus reporter particles that these are in fact sort of cell type specific loss of tropism mutations. So after we confirmed using these pseudoviruses and reporter particles that these were indeed loss of tropism mutants, we then engineered them into replicative chikungunya virus because we have strong evidence, which I just showed you, that these would be loss of function rather than gain of function mutations. And so here's now showing the growth of replicative chikungunya virus with these different mutations. And you can see that the replicative chikungunya virus with the human-specific loss of tropism mutations indeed have reduced, although not totally eliminated, growth in mixer 8 expressing human cells. And conversely, the ones with the sort of mosquito loss of tropism mutations have reduced growth in the mosquito cells. So sort of validating that we can actually use this information to design chikungunya virus mutants that have lost their ability to infect at least efficiently, both human and mosquito cells and have sort of had their tropism reduced to just one of those species. So to sort of conclude, I described how we measured how all mutations to the chikungunya virus envelope proteins affected entry into 3 different cells. Although we still do not know the mosquito receptor, we identified which mutations specifically impair human versus mosquito cell entry. And obviously, we're interested in ways to use that knowledge to discover what the mosquito receptor is. So if people have ideas, let us know. We are going to try sort of differential mass spec pull down with the mutants that sort of can and cannot enter mosquito cells. And we're also interested in the idea of whether we could somehow use this information in combination with like AlphaFold 3 to predict it. But if anybody has a lot of expertise in that, let me know. But these turn out to be, although AlphaFold 3 is great, these turn out to be big, complicated proteins. And AlphaFold 3, at least in the vanilla form, doesn't even correctly predict the binding of the envelope proteins to mixerate, even though that structure is actually in the PDB that was used as a training data set for that. And I think it's because these are such big, complicated proteins with complex information. We use this information to rationally design chikungunya virus mutants that could only efficiently infect human or mosquito cells. And then we think these loss of tropism mutants are potential vaccine candidates. There is a live attenuated chikungunya virus strain that is being explored as a vaccine and used in some cases, but it does occasionally have sort of moderate to mild side effects. And in particular, these loss of function mutants should have lost their ability to actually transmit by any sort of natural transmission cycle. And then also, we think all this data about the effects of mutations to these proteins could be useful for engineering non-replicative, like nanoparticle and so on, immunogens. Then finally, all the data I described here is available on this big web page if you want to go look at it more. And I'd like to thank, in particular, Zhao Wei, who's a postdoc in our lab, who really led and did pretty much all the experiments I described, as well as Will, Kalen, and Brendan in our lab, which helped out with certain aspects of the analysis and experiments. And then we also really benefited from collaborating with some researchers at Washington University as part of this REVAMP network, where we're working on alphavirus vaccines, in particular Mike Diamond, who I'd never collaborated with, but is an amazing person to collaborate with and really helped us get all of this going in our lab. And I'd be happy to answer any questions for the last few minutes.